Hello everyone, this is Victoria and welcome to the second video in the series in which I explain the book called Principles of Anatomy and Physiology. So today we are going to talk about Chapter 2, the chemical level of organization. So in this chapter we're going to talk about how chemistry makes up our body and how it helps us perform many important functions. So let's get started with the mind map. There are 5 main things that we're going to talk about today. Firstly, we're going to talk about matter, the major and minor elements that compose our body. We're going to talk about the importance of the electron and how things such as isotopes and free radicals contribute to the building of our body. Secondly, we'll talk about the different types of chemical bonds, mainly ionic, covalent, as well as hydrogen bonds. Then we'll be looking at chemical reactions, specifically types of energy, types of chemical reactions, and we'll look at some specific examples for each. Then we're going to talk about the inorganic compounds in our body, which specifically we'll be spending some time on water, some of its properties, and why it's especially important for us. We'll talk about hydrolysis and dehydration synthesis, which are 2 important types of reactions that water supports. We'll talk about mixtures in our body and also pH as well as buffers. Last but not least, we're going to talk about the 4 main types of organic compounds in our body, which are carbohydrates, lipids, proteins, and nucleic acids. And we'll also talk about ATP, which is the main energy currency. So let's get started. Talking about matter. Our bodies are made of 26 different elements, 4 of which are called major elements, and they make up 96% of our body. Specifically, they are oxygen, carbon, hydrogen, and nitrogen. These are very essential for us. We have also 8 lesser elements, and they are calcium, potassium, sulfur, sodium, chloride, manganese, iron, as well as phosphorus. We also, so these lesser elements, they make up 3.6% of our body, and the rest 0.4% are made of what we call trace elements. There are 14 of them, and we only require them in small amounts, and they are aluminum, boron, chromium, cobalt, copper, fluorine, iodine, manganese, molybdenum, selenium, silicon, tin, vanadium, and zinc. So here let's take a look at what atoms are. So atoms are the smallest unit of matter that retains the property and characteristics of an element. So here we can see that there are 3 main subatomic particles that make up an atom. They are protons, which have a positive charge, neutrons, which don't have a charge, and electrons, which have a negative charge. We see here in the graph that protons and neutrons make up the nucleus of an atom, and electrons are distributed outside the nucleus. So we have 2 models that tell us how atoms potentially look like. We have the electron cloud model, which says that electrons are present in cloud-like shapes outside of the nucleus. And we also have the shell model, which indicates orbitals that the electrons should be in. So there are a few terms that would be useful to know in chemistry as well as throughout this book. We have an atomic number, which is what defines an element. So this is based on the numbers of neutrons that an atom has. So how many neutrons an atom has indicates which element it is. Then we have the mass number, which we can understand it as how heavy this atom is. This is the number of neutrons plus the number of protons, because these 2 subatomic particles have weight, while electrons are really, really light, so we neglect their weight. Then we also have something called the atomic mass. So it is the average mass of all the atoms present in nature. So it is different from mass numbers. So a mass number is always an integer. But an atomic mass is the number you see under periodic tables, and it can be numbers like 11.99, and these indicate this is an averaged out of the mass of different isotopes. So isotopes are of the same element. They all have the same number of neutrons, but isotopes differ from each other in that they have different numbers of protons. So an example is we have C12, which has 6 neutrons, 6 protons. We can have C13, which has 6 neutrons and 7 protons. And we also have C14, which is 6 neutrons and 8 protons. So they all have the same number of neutrons, showing us that they're the same element. But they have different numbers of protons, which contribute to different mass numbers. There are also, in isotopes, there are also what we call radioactive isotopes or also radioisotopes. So they're quite unstable and their nuclei sometimes decay or spontaneously change into more stable configurations. So for example, C14 that we just mentioned is a radioactive isotope. So when these isotopes decay, they emit radiation, which we can understand it as packets of energy. And these can sometimes be harmful for us in large amounts, but they can also help us measure, for example, how long this fossil has existed. With isotopes, we have half-lives, which is the time it takes for half of the isotope to decay. Some other things useful to mention when talking about matter are ions, molecules, and compounds. So it would be beneficial to clarify the differences between them. So when an atom, which has a neutral charge, gives up or gains an electron, it becomes an ion. So it either carries a positive charge, meaning it has more protons than electrons, or it carries a negative charge, meaning it has more electrons. Then we have what is a molecule, which is when two or more molecules combine together. So they do not necessarily have to be of the same element. They can be of the same element or a different element. For example, O2, oxygen, it is called a molecule. But we can also have things like H2O, like water. They are also called molecules. So it's made of 2 or more atoms. Then we have a compound. A compound is a substance that has atoms of 2 or more different elements. So the elements must be different. So as we just mentioned, oxygen is not a compound because it only has one element, but water is a compound. Lastly, we see here in the mind map that we have something called free radicals. So these are atoms or groups of atoms with unpaired electrons in the outer shell, the outermost electron shell to be clear. So an example is called a superoxide. And here is what a superoxide looks like. We can see here that in the outermost shell of the oxygen to the right side, there is a lonely electron that is not paired. And these superoxides or just free radicals in general are very unstable and reactive, and they can be destructive to nearby molecules. They grab away the electrons or they give away their electrons to nearby molecules, and they may break apart important bulky molecules, which is why we want to avoid accumulating too much free radicals in a body. Something that counteracts free radicals is called antioxidants, which inactivate these oxygen-derived free radicals. So now let's talk about chemical bonds. So before we dive into the different types of bonds, let's talk about the octet rule. So we have mentioned how atoms, they have electron shells. The octet rule basically states that every atom is most stable when it has 8 electrons in its outermost shell. So let's look at this with an example. First, let's examine ionic bonds. So we see here we have sodium atoms and chlorine atoms. In our sodium atom, we can see that there is one electron in the outermost shell, which makes it reactive. If we want the sodium to become stable, we would want to have 8 electrons in its outermost shell. So we have two options. Either we give out the electron in the outermost shell, or we have to gain 7 more electrons. So obviously it's intuitive that giving out 1 electron will be easier than gaining 7 electrons. So this is what sodium would do. It would donate 1 electron, and it would gain a positive charge because now there are more protons than electrons. So now sodium is an ion, and the opposite thing happens with chlorine. So we see that chlorine has 7 electrons on its outermost shell, and to satisfy the octet rule, it would want to gain 1 more electron. So it takes the electron that sodium has given out and absorbs it into itself. So it now becomes a negatively charged ion. Also, one thing that you should know is that negatively charged ions are called anions, and positively charged ions are cations. So now we just saw the process of how sodium gave an electron and chlorine grabbed the electron. So this process of transferring an electron from one substance to another substance is called basically just makes a bond that we call ionic bond. And this is usually between metals and non-metals. Next, we move on talking about covalent bonds. So covalent bonds are the sharing of electrons. They do not involve transferring electrons. Let's look at these with some examples. So first, we see that let's look at the second example with oxygen atoms because it more strictly follows the octet rule. Oxygens have 6 electrons in its outermost shell, or 6 valence electrons, and it would want 8 electrons in its outermost shell. So if the 2 oxygens share 2 electrons in its outermost shell, we can see that they can have 8 electrons. So basically what they do is they share the control of 2 electrons. So they have 6 electrons of their own, and they are also having, we can say, control of 2 electrons from the other molecule. So together that makes 8 electrons in the outermost shell. So we see that here there are 4 electrons shared between the oxygen molecules, which makes up a double bond. Here from hydrogen, there are 2 electrons being shared, which makes up a single bond. And here in nitrogen, we see that 6 electrons are being shared, which make up a triple bond. So we count these bonds in the number of pairs of electrons that are shared. We see that covalent bonds can also give us more complex structures, such as the methane molecule we see here, as well as the water molecule down here. So with covalent bonds, we also have to consider whether an atom is polar or not. So different atoms, they want electrons in different degrees. For example, oxygen wants electrons more than nitrogen because it only needs 2 electrons to satisfy the octet rule, while nitrogen needs 3, so it wants to grab electrons more. Another term for this wanting to grab electrons is called electronegativity. So oxygen has quite a high electronegativity, while atoms like hydrogen only have a middle negativity. They are fine to give an electron, they're also fine to grab an electron. So when atoms want electrons differently, they have this, you can understand it as this push or pull conflict. So let's look at the formation of the water molecule. The oxygen really wants the electrons, so it tries to grab the electron and hold the electrons to its side. While the hydrogens are like, okay, they sort of give up fighting with the oxygens. So even though we think that the 2 hydrogens and the oxygen are sharing their electrons, actually the electrons are spending more time by the oxygen atoms, which make the oxygen slightly more negative. So we can see here These signs indicate that oxygen has a partial negative charge and that hydrogen has a partial positive charge. And these atoms with these partial charges are called polar molecules, and they are important in their properties in respect to water, which we'll also mention later. And these first four molecules that we see here, they're all examples of nonpolar molecules because they're sharing electrons equally between them. And in the case of methane, even though carbon slightly wants more electrons than hydrogen, we see that they're in a symmetrical arrangement. So these partial charges, they also balance out each other. So the last type of bond that we'll talk about today is the hydrogen bond. So here is a graphical illustration of a hydrogen bond. So they occur between hydrogens and other atoms such as oxygen or fluorine. So as we mentioned before, when hydrogen forms covalent bonds with other highly electronegative atoms, they gain a partial positive charge, and the other atom, such as oxygen, gains a partial negative charge. We all know from physics that positive charges and negative charges attract each other. So when we have partial negatives and partial positives, they also form this type of attractive force that pulls the different atoms together. So the hydrogen wants to be closer together to the oxygen of other water molecules, and the oxygen in our water wants to be closer to the hydrogens of other water molecules. So this is a type of bond that pulls different atoms together, and this also contributes to some of the unique traits of water that we'll mention later. So now let's talk about chemical reactions. So in every chemical reaction, we have our reactants and our products. So the reactants interact in a way to form our products. One example is the production of water. So we have two hydrogen molecules and one oxygen molecule, and they react together to form two water molecules, which is our products. Then we have two different forms of energy that would be useful for you to know. We have potential energy, which is energy stored by matter due to its position. And we also have kinetic energy, that is energy associated with motion and movement. So one typical example is a ball on a hill. So when the ball is on the top of the hill, it has a lot of potential energy because it's high above the ground. There is the gravitational potential energy. And when the ball starts to roll down the hill, this potential energy gets converted into kinetic energy because it is moving. And in chemistry, chemical energy is a form of potential energy that is stored in the bonds of compounds and molecules. We also have something important called the law of conservation of energy, which tells us that energy can neither be created nor destroyed. It can only be converted from different forms into other forms. Then we have two types of chemical reactions. We have exergonic reactions, in which the reaction overall, the net reaction releases energy. So it is giving out energy into its environment, which can be in the form of light or, for example, heat. And in contrast, we have endergonic reactions. So it is absorbing energy from its environment more than it is releasing. So overall, it's taking in energy from the environment to conduct the reaction. And then we have our activation energy. So I think this graph would be helpful to understand. First of all, we can see we started with more energy in the reactants and ended with less energy in the products. So before we go directly from the energy of the reactants into the energy of the products, we have to go through a peak, and the height of this peak is called the activation energy. So this is basically the minimal amount of energy that you need to break the chemical bonds so the reaction itself can start. So without the sufficient activation energy, the chemical reaction won't start, even if you have all the reactants. So taking the example of the formation of water that we talked about, in order to have H2O, we have to break the bonds between the hydrogens. We also have to break the bonds between the oxygens. If we don't have the sufficient energy to break the bonds, we can't have these raw materials for us to make a water molecule. So this is the investment that we would need to start a reaction. And then the formation of new bonds might also release energy into the surroundings. And then what catalysts do is that catalysts lower the activation energy. So in the second graph that we can see, the blue curve is the activation needed with the catalyst. So when you need less activation energy, it's easier to start a reaction. So this is why catalysts often speed up reactions. And in the case of us, of living beings, living organisms, our biological catalysts are called enzymes. So this would be helpful to just know. And then lastly, we look at some of the types of chemical reactions. First, we have synthesis, in which different atoms or molecules come together to form a new molecule, which is, for example, what we talked about in water formation. Hydrogen plus oxygen gives us water. And then we have decomposition reactions, which is breaking down. So we have a molecule, for example, AB, and it breaks into A and B. One example is methane. So methane is CH4, and it breaks down into one carbon as well as two hydrogen molecules. Specifically, when we perform these synthesis and decomposition processes, we call them anabolism and catabolism. So a synthesis reaction is anabolism, in which we typically, these connecting to what we talked about before, anabolic reactions are typically energonic because they're absorbing more energy than they release, and they combine simple molecules into larger molecules. We also have catabolism, which is the case of decomposition reactions. They are usually exergonic because they are releasing energy they're releasing more energy than they have absorbed. So for example, we break down big molecules like glucose and we produce smaller molecules with them, and this is also the mechanism through we can get energy in our body, which we'll also talk about in later chapters. Then we have what we call exchange reactions. So this term is different from what we might have conventionally learned in chemistry, which is called displacement reactions. But basically, the reactants switch partners. So originally in our reactants, we have AB and CD. And after we complete the reaction, we have AD and BC. Or for example, AC and BD. So basically, the reactants, they switch a partner. So for example, this is typical of an acid-base reaction. If we had hydrochloric acid, which is HCl, plus sodium bicarbonate, for example, which is NaHCO3, then they switch partners. So the chlorine in the hydrochloric acid goes to the second reactant. It goes to the sodium to form sodium chloride, or salt as we know it. And the H in the hydrochloric acid combines with the HCO3 in the sodium bicarbonate, and together they form carbonic acid, which is H2CO3, just for your information. So basically, the ions in our compounds have switch partners, and this is called an exchange reaction. Then we have reversible reactions, which is a type of reaction in which the products can go back into their original reactants. So we have the reactions going into both ways. So water also applies. After we formed water, it can also heating it and flowing electricity through it also allows us to break water back into hydrogen and oxygen. So there are also reversible reactions that occur without too much energies, and we'll talk about these in later chapters of the book. And lastly, we have redox reactions, which is also called oxidation reduction reactions. So so the RED is from the reduction part, and the OX is from the oxidation part. So oxidation refers to the loss of an electron, and in the process, the oxidized substance loses, it gives out energy, and the reduction is the gain of electrons. So in the process, the reduced substance gain energy. So redox reactions are always accompany each other. So whenever we have a substance being oxidized, which gives out its electron, something has to take in the electron. So these always go hand in hand. Whenever we have oxidation, we also have reduction. So this will be more specifically covered when we talk about how we break down food, how we oxidize substances like glucose to give us energy. Now let's talk about some of the main inorganic compounds that form our body. So firstly, we have water. As you must have heard your parents and your teachers talk a lot about, it's important to stay hydrated, and the reason is that more than half of our body mass is made up of water. And there are some properties of water that make it especially important. So firstly, as we mentioned, water is a polar molecule. The oxygen of our waters they carry a partial negative charge, and the hydrogens carry a partial positive charge. So this makes water a very versatile solvent because it can bond with a lot of ionic and polar substances. So here, this graph is useful in helping us illustrate why water is good at dissolving things. So it can form a lot of polar covalent bonds, and because of these partial positive and partial negative regions. For example, we see that the oxygens, which are partially negative, are all facing our sodium ion, and this convinces the sodium ions to break apart from the chloride to be surrounded by the waters. And we also have our chloride ion, which are surrounded by the partially positive hydrogens. So this bent shape of water also helps it to form a lot of bonds with other ionic or polar substances. So this is why it is called a universal solvent. It's really good at dissolving a lot of polar compounds. Substances that are charged or substances that contain polar covalent bonds are called hydrophilic substances because they combine with water and dissolve easily. We also have, however, hydrophobic substances that are not really soluble in water, and these are usually nonpolar substances. Then some of the other properties we have are water. Then we have some of the thermal properties of water. First of all, it has a high heat capacity, meaning that water can absorb and they can release a relatively large amount of energy with only a modest change in its own temperature. So it takes a lot of energy to really boil water. So this is what we refer to through a high heat capacity. And this allows us, for example, this is like how sweating works. So water vaporizes, and when it vaporizes, it carries a lot of heat out of our body. So this is why heating actually helps us to cool down. And water also acts well as a lubricant. So this is why it is a major component of mucus and other lubricating fluids throughout our body. It can, basically, as the word itself suggests, it can be between spaces and help make processes physically more smoother. And there are two types of chemical reactions that are especially important and involve water. First, we have hydrolysis. So hydrolysis enables nutrients to be absorbed in the body. So when we add water to a particular substance, it breaks larger nutrient molecules into smaller molecules when we add water. So this is also why we call it hydrolysis, because we're adding in water to break things. Then we have dehydration synthesis. So from the name, we know that we're making some molecules and we're getting water out of it. So when two small, this is usually happening when we join two smaller molecules into a larger molecule, and water is a byproduct of this process. So this is also a way that a lot of polymers are formed from monomers, which we'll also cover later. Then we will briefly distinguish between mixtures, different types of mixtures. So mixtures are basically a combination of elements, compounds, and things, and they are physically bonded together, but not by chemical bonds. And then some of the different types of mixtures that we have solutions, which are homogeneous, so they mix together really evenly. So for example, we have salt and water. This is a solution. We have colloids. So colloids are different from solutions because the size of its particles. So in colloids, the solid particles are really large and they can scatter light. So for example, it's like, for example, consider fog. And the water molecules in the fog are large enough, so when a car goes through fog, and we can see the headlight scatter the water particles in the fog. So this is a colloid. And last, we have a suspension. So in the suspension, the solutes do not settle out and they accumulate on the bottom of the container. So this is a suspension, so the solute is not actually dissolving with the solvent and it settles out eventually in the bottom of whatever is the container. Then moving on, we talk about pH, which is a really important thing to mention when we're considering solvents and solutions. So first of all, we need to distinguish between acids and bases. So usually acids are things that give out H+. They are also called proton donors. They give out our hydrogen ions, and they usually taste sour, but please don't try that. And we have bases. Bases, they take in usually the H+, our hydrogen ions, and in a lot of cases they have hydroxide ions, so they have OH-. The defining characteristic is they are able to remove the H+ in the solution, and they are called proton acceptors. We also have salts. So when we dissolve salts in water, or also some of other solvents, they dissociate into cations and anions, which are not hydrogen ions or hydroxide ions. So for example, we have sodium chloride, which is what we also know as salt. So it dissociates into our sodium ion as well as our chloride ion. So acids and bases, as we talked about, often react, and they perform exchange reactions. So another example we have is hydrogen chloride plus, for example, sodium hydroxide, and this would give us sodium chloride as well as a water molecule. So the common reaction equation for acid bases is an acid plus a base gives us a salt and some water. Then we get to the concept of pH and pH scales. So pH basically is the concentration of hydrogen, and the equation of pH is negative log the concentration of hydrogen ions. So we express how acidic or how basic a solution is with a pH scale on a scale of 0 to 14. So a pH below 7 means that a solution is acidic and it contains more hydrogen ions than hydroxide ions, and a pH above 7 indicates the solution is basic. So it has more hydroxide ions than hydrogen ions. And a solution, a pH of 7 is considered neutral, so it has the same amount of both. So although the pH of body fluid often differs and varies sometimes, the normal limit of the pH of our fluid is quite narrow, and it's important for our survival that we control the pH of our blood so that it's not too acidic and also not too basic. for this, we have what we call buffer systems. So in a buffer system, when we add an acid to, for example, a neutral solution, it doesn't change the pH so much. So if we directly add, for example, hydrogen chloride to a solution, the pH might drop to something like 4 or 3, but if we add it to a buffer system, it only changes like maybe by 0.1 or something like that. So it tries to maintain the pH around this constant neutral scale and help it not fluctuate too much. So it achieves this through having acids and conjugate bases. So when we add an acid, it reacts with the conjugate base and it sort of neutralizes. And when we add a base, it reacts with an acid, so it also neutralizes. And these adding are all reversible reactions that we talked about, so it can go both ways. So this helps to neutralize most of the acids and bases being added. And in our body, one of the important buffer systems we have is the carbonic acid bicarbonate buffer system. So we briefly talked about carbonic acid and how it's created with carbon bicarbonate previously. So this our carbonic acid acts as weak acid, and the bicarbonate ion is the weak conjugate base that we talked about, and it compensates for an excess or in shortage in hydrogen ions with the mechanism that we discussed. Now let's talk about some of the organic compounds making up our body. So one key thing about organic compounds is that they have a lot of C, H, and O, carbon, hydrogen, and oxygen, and these are the main elements making up organic compounds. And some other elements also have nitrogen, phosphorus, and sulfur. Carbon is usually the backbone of our organic molecules, meaning it is throughout the molecule. There are 4 main macromolecules that we usually talk about. Firstly, we have carbohydrates. So we need here to mention another concept, that is monomers and polymers. So a monomer is the building block of our macromolecules, and when we join a lot of such monomers together, we get a polymer. So as we talked about before, we have dehydration synthesis, which is a method of joining monomers to form polymers. And let's talk about carbohydrates first. So the monomer is monosaccharides. So from the prefix mono, we know that it is a single unit. And they're usually ring-shaped carbons. And we have, for example, glucose, which is our blood sugar. We have fructose, which is in fruits. We have galactose, which is found in sugar. And then disaccharides, from the prefix di, we see it has 2 monosaccharides combined together by dehydration synthesis. For example, we have here glucose plus fructose. It gets us sucrose, which is what we see in table sugar. And this graph also illustrates the role of water in forming these monomers as well as polymers. So monosaccharides are the monomers of carbohydrates. The polymers are called polysaccharides, which is also quite self-intuitive by its prefix. So some examples of polysaccharides include glycogen, which is the form of storing carbohydrates in animals, starch, which is the form of storing carbohydrates in plants, and also cellulose, the tough part of cell walls in plants that cannot be digested by humans. So carbohydrates usually represent only 2% to 3% of our bodily masses, and they mainly function as a source of chemical energy. Then let's talk about the second class of macromolecules, which are lipids. So lipids are also what you think of as fats and oils. So the monomer of lipids are fatty acids. So let's take a look at this triglyceride here. We see this this tail part where you have a long carbon backbone and a double bonded oxygen attached to it, this is what we call a fatty acid, which are also the gray parts shaded in the graph. So fatty acids can either be saturated, meaning it is all single bonds, or unsaturated, which we see in the bottom fatty acid. So it has a double bond in our carbon chain, which gives it a kink, and this kink just tells us basically this is a unsaturated fat, and this is useful in terms of their reactivity. So fatty acids are the quote-unquote monomers. They're not true monomers because we only see normally 3 or 2 fatty acids making a macromolecule, but they are the basic, we can understand, a building block of most lipids. Examples of some lipids we have is triglyceride, as shown here. We have 3 fatty acids, and we have 3 fatty acids here. And another really famous example of lipids is phospholipids. So it is present in our cell membrane. We have the phospholipid bilayer. So what is special about a phospholipid is that it has a polar head due to its phosphate groups, and then it has 2 nonpolar fatty acid tails. So in the cell membrane, the tails are facing each other and the polar heads face outward or face the solution outside of the cell or inside of the cell. So this is really important in making up our cell membranes. Another type of lipids that we have are steroids. So some steroids that might surprise you include cholesterol, some salts, and bile. Vitamin D is actually also a type of steroid, and we also have some hormones. So steroids are different from these in terms of mainly their structure. They are more like carbon rings with attached fatty acid tails. Then let's talk about proteins. So proteins are long chains of amino acids. So amino acids are the basic monomer of proteins, which is the polymer. Taking a look at the basic structure of amino acids, so we see here, first of all, we have a central carbon, which is attached to a hydrogen, a amino group, and a carboxyl group. So one is basic and one is acid. And we also have a R group, which is also called the side chain. So this is what defines each protein. So these proteins have different properties, they have different functions, they have different names because of the different side chains that they are attached to. And they have different types of charges, etc. And also, we see here that in the amino group, we have 2H, and in the carboxyl group, we have an OH. So when we want to combine amino acids together, the H in our amino group as well as the OH in our carboxyl group, they actually combine together, and we have dehydration synthesis again. And the bond that they form is called a peptide bond. When we talk about proteins, we also talked about the 4 levels of structure in a protein. So the primary structure in a protein is the sequence of amino acids. So we have different amino acids linked together in a specific sequence that define our proteins. The secondary structure is basically how amino acids interact, how they form, for example, hydrogen bonds, how they twist and fold and form different bonds. So this is basically some of the shapes that they fold into. The tertiary, also just to mention, in secondary structures, 2 common shapes that we see are alpha helixes as well as beta plated sheets. Then in our tertiary structure, we see like the 3D shape of the whole protein, like how the whole polypeptide chain is folded. And in a quaternary structure, we have 2 or more such polypeptide chains. So we have individual polypeptides that folded into their shapes, and these polypeptide chains also interact with each other. So these are quaternary structures. And proteins are really essential for us when we think about enzymes. So enzymes are mainly proteins, and the shapes that they fold into allows them to perform different specific functions. Then we have our nucleic acid. The monomer of a nucleic acid are nucleotides, and this macromolecule does not give us any energy. It is important. It holds our genetic material. So let's take a look at what our nucleotides look like. First of all, we have a sugar. So the sugar is different in DNA and RNA in terms of the presence of an oxygen that we can see here. So this is why one is called deoxyribose and one is called ribose. Then the pentose sugar is connected to two things. Firstly, we have a phosphate group. So together with the phosphate, DNA has a sugar phosphate backbone, which forms basically the two backbones of a twisted double helix, or also the single helix in RNA. Then it is attached to a nitrogenous base, which quote-unquote defines the identity of our nucleotide. So there are 5 possible nitrogenous bases. DNA has ATCG, which are adenine, cytosine, thymine, as well as guanine. And in RNA, we don't have thymine. Instead, we have uracil. So we don't have T, we have a U. They're different in terms of their structure, and we also see they're classified into pyrimidines, which only have 1 ring carbon, and purines, which are bigger and have 2 rings of carbons. So whenever they pair together, A with T, C with G, a pyrimidine is pairing with a purine, and they form double bonds or triple bonds. So the sequence of how these ATCGs are connected codes the genetic information of living beings. So this is also a very important aspect. Last but not least, we have ATP. So we all know that ATP is the energy currency of our bodies. However, ATP is not a macromolecule, but it's still quite important to talk about. First, let's take a look at its structure here. The full name of ATP is adenosine triphosphate. So we can see in the graph clearly, the adenosine refers to an adenine group and a ribose sugar. And then we have 3 phosphate groups in ATP. And the last phosphate group, the third phosphate group, is what gives it energy. So when we break off the phosphate group, it gives off energy and it converts to ADP, which is the unenergized form of ATP. So what is really important and what you should take away from this is that the third phosphate group is what is essential in the structure of ATP and what powers our whole body. So, yeah. So that's about it for today. So we talked about, we mentioned different types of matters and the elements present in our body. We talked about free radicals. We talked a little bit about the structures of atoms and what defines different elements. Then we covered the 3 main types of chemical bonds, as well as how they abide by the Octet rule. We mentioned chemical reactions and different types of chemical reactions in terms of their energy, in terms of the process of their overall reaction, as well as how the reactions are interacting to form what types of products. Then we mentioned the main inorganic compounds in our body, specifically talking about properties of water and why it's essential to us. We talked about different types of mixtures, and we also talked about pH and the logic behind buffers. Last but not least, we covered the 4 main macromolecules in our body, which are also organic compounds. And we also talked about ATP, which is the energy currency in our body. So I hope you learned something from today's video. Again, thanks a lot everybody for watching this video. I hope you learned something about the chemical level of organization of our body. In the next chapter, we'll talk about the cellular level of organization. We'll be talking about different types of cells, some structures present and their functions, how cells communicate, etc. So we'll be diving into the cellular level. If you're interested, stay tuned in. If you enjoyed this video, please give me a like and feel free to give me any comments. Yeah, and see you next time.